हेलो फ्रेंड्स, वेलकम बैक टू बायोफाइल लाइफ क्लासेस। माय नेम इज मोनिका शर्मा और आज हम पढ़ने वाले हैं प्रोकैरियोटिक सेल्स के बारे में। बट बिफोर वी बिगिन टुडेज डिस्कशन, आई वुड लाइक टू टेल यू that this video is the part of lecture series that we have been doing for chapter 8 Cell, the unit of life, class 11 Ncert biology. The part 1 has already been uploaded, and in that video we had already discussed about the basic features of a cell and cell theory. The link for the same has been provided in the description box. Kindly go through it before you come to this part 2. So in this video we'll study about prokaryotic cells. So what is prokaryotic? The word prokaryotic is made up of two words, pro and karyon. Pro means primitive and karyon means nucleus. So these are prokaryotic cells are those cells which have nucleus present in their primitive form, and these primitive form nucleus are found in early living forms. So primitive nucleus means the nucleus is not well defined. तो यहाँ पे बेसिकली आप देख सकते हैं इट्स अ पिक्चर ऑफ अ टिपिकल प्रोकैरियोटिक सेल वेयर दिस पर्पल कलर थिंग इज अ न्यूक्लियस, and we do not call it a nucleus. Let's call it genetic material, because for calling something as nucleus, it has to have some certain characteristics which are not found in the genetic material of a prokaryotic cell. So in this this purple color thing is the genetic material found in a prokaryotic cell. As you can see, it is loosely bound. It is not well defined, and it is not covered by a well defined nuclear membrane as well. And the place where it resides inside a prokaryotic cell, that region is known as nucleoid. So if somebody is asking you what is nucleoid, you need to say that nucleoid is the region within a cell where the genetic material resides. So you can see this nucleus is in primitive form. Now you can find there is no well defined cell organelles. These blue dots are some of the micro bodies which are found, that we'll be studying later in the video. And now this prokaryotic cell is covered by a cell wall. The outermost layer is a cell wall, then a plasma membrane and a capsule. And there is a flagella present. This flagella is used for its movement, and there are pilus present all over the body which are useful in the conjugation process. So once these are the basic features, you need not to be worried about it right now. But so let us now study the basic features of a prokaryotic cell. The important features are such like that. These are the cells which are found or which are represented by the bacteria, the early forms. These are the early forms: blue-green algae, mycoplasma and PP yellow. They are smaller in size, there can be single cell also, and they multiply very rapidly. They also vary in shape and sizes. So basically if you see any bacteria, the basic four forms in which the bacteria are found can be bacillus, that is rod shaped; coccus, that is spherical in nature; spirillum, the word itself says it is spiral; and the vibrio is comma shaped. So these are the four basic forms in which the prokaryotic cells exist. The cell membrane is surrounded by a cell wall, there you can see, and this exception you need to remember: in case of mycoplasma there is no cell wall. The nucleus is not well defined, that is why we designate it with the word called nucleoid. So nucleoid is basically a region where genetic material is present. There is no well defined nuclear membrane, and some prokaryotes, they have a circular DNA which is outside your genomic DNA. So genomic DNA is present in nucleoid, and outside that genomic DNA there is some other circular DNA present. These circular DNA are called plasmids, and these plasmids are very much having its applicability in genetic engineering and all. So that is, we will be doing in other chapters what is the use of this plasmid, and there are many things about it. For now, you need to focus on very basic features of a prokaryotic cells. Now come to ribosomes. Ribosomes are the cell organelle which are present both in the eukaryotic cells and prokaryotic cells. The only difference is that the type which is present in prokaryotic cell is known as 70S type, and the thing which is present in eukaryotic cell, that is called as 80S type. That will be doing. So what is 70S type prokaryotic ribosome? So 70S ribosome, it has two subunits. The larger subunit is called 50S and the smaller subunit is known as 30S. During the process of translation, these two combine and they help in the protein synthesis. So for now, you need to remember that ribosomes are the site for the protein synthesis. 70S type is present in prokaryotic cells. The 70S is made up of two subunits. The larger subunit is 50S and the smaller subunit is 30S. When many ribosomes, they come together and they attach to mRNA to undergo translation process, then that thing is called polyribosomes or polysomes. So what is translation? I hope you are familiar with the concept of central dogma. So how central dogma works? It's from DNA to mRNA, and from mRNA it is leading to proteins. So the process from conversion of the DNA and coding from DNA to the mRNA, that process is known as transcription, and from mRNA to protein formation, that process is known as translation. So ribosomes basically help in the process of translation, that is from mRNA into the proteins. The next part which is present in the prokaryotes and is one of the characteristic feature of a prokaryotic cell is called mesosome. As you can see, this is the cell membrane, the outer wall is the cell wall, and there is an infolding, an extension in the cell membrane which is called as mesosome. So as you know that prokaryotes do not have well-defined cell organelles like endoplasmic reticulum or mitochondria. So therefore, in the absence of those, the process of transportation, the process of respiration in prokaryotic is done by the cell organelle called mesosome. So this is what a cell mesosome looks like. So basic features are, a mesosome is nothing but an infolding of a cell membrane. It helps in the increase in the surface area, and thus it helps in the process of respiration. It also helps in transportation as well. The next part is, this cell exists in itself also. So there can be single celled prokaryotic cell organisms also. So for this cell to move here and there, it should have certain filaments which help a prokaryotic cell in movement. So one such filament is known as flagella. So flagella are nothing but the thin and filamentous which originates from the cell membrane. Here you need to be aware that it is not originating from the cell wall, but the flagella is originating from the cell membrane. So it is originating from the cell membrane. It can be one or it can be multiple, and that you need not to go through in detail. And what happens, this is how the movement of the flagella looks like. It's like a whirlpool. So when it is moving like a propeller like motion, it makes the cell to make a movement, and that is how a cell moves. We also call it whip-like movement, and this flagella, why the name flagella is given, because it is composed of flagellin protein, and the sole purpose of a flagella is the motility, that is the movement of the prokaryotic cell. The other is the cilia. In some prokaryotic cells, you can find the presence of cilia. Now the difference between a flagella and a cilia is, flagella can be one and the movement is like a propeller like, but in case of cilia, the cilia is always more than one. So it can be a pair in it can be present in the pairs, it can be present all over the surface of the prokaryotic cell and such, and the movement is always synchronous, and it is forth and back beating of this cilia which helps in the movement of the bacteria. So back and forth beating movement is restricted to cilia, and propeller-like motion can be seen in case of flagella. However, both flagella and cilia are the cell organelles, are the protein-based filaments which are going to help the cells in their motility. Now, apart from motility, there is another protein-based filament present, which is known as pili. These pili is also present all over the surface of the cell, and these help in the process of conjugation apart from the process of motility. So in case of pili, you can see these are hair-like non-helical appendages. These are straight, they are not helical like flagella, and they originate from cell wall. In case of flagella, it was originating from the cell membrane, that is plasma membrane. But in case of pili, these are originating from the cell wall. These are composed of pilin protein. They are numerous in number and they are present throughout the cell wall. The main function of a pili is in the process of conjugation, but it also helps in the motility. So I hope these things are clear with you. Now, prokaryotic cells, as I already have said in previously, that prokaryotic cells, they lack the well-defined cell organelles. For example, mitochondria, endoplasmic reticulum, Golgi apparatus. These are the cell organelles which are found in eukaryotic cells but not found in prokaryotic cells. And apart from these, inclusion bodies are also present in the cytoplasma of a prokaryotic cell, which is not membrane bound, and it lies freely in the cytoplasma. The thing which I have shown you in the first slide, the blue dots which are represented by ribosomes, apart from the ribosomes which are freely available, there are many inclusion bodies as well. And these inclusion bodies can be phosphate granules, cyanophycein granules, glycogen granules, that is the food. Gas vacuoles are present in blue-green algae and purple and green phytosynthetic bacteria. So once you are done with prokaryotic cells, I want to draw your attention to one technique which is given in your book. It is called gram staining. So what is gram staining? You know, there are millions of bacteria and the varieties of bacteria found all around us. So bacteria are basically, we are on the basis of the cell membrane or the outer layer of their cell wall. We are dividing the bacteria into two large groups, that is gram positive bacteria and gram negative bacteria. This technique is very easy and is very common, and you can easily do it in your biology labs. I don't know if you are doing it right now currently in your bio labs or not, but for us it was a very common technique. So why gram staining? Staining means you need to color it, and gram is the word is given by it is for the name of the person who actually devised this technique, and the name of the scientist was Christian Gram. That's why the name is gram staining. So what we are doing, we are going to distinguish between a gram positive and a gram negative bacteria. And what is the principle behind it? The principle is, some bacteria have a very thick layer of peptidoglycan in their cell walls, while other group has very thinner peptidoglycan cell wall. So based on this difference, the cell wall is going to react or going to stain to the colors, and on the basis of retaining the stain or retaining the color, we are dividing them or distinguishing them between gram positive and gram negative. Very quickly I am going to tell you what exactly we do it. Here we have a primary stain. Stain means color. For now you need to understand, stain means color or dye or whatever you call it, it's a color. So you are going to use a crystal violet color. Naturally the color of this stain is a violet. So you are now, once you have taken a cell, you put a drop of crystal violet on it. That cell is going to retain that, is going to be in violet in color, and after that you are putting iodine solution, that is grams iodine. This is the main this is the main ingredient in this technique. So when you are using grams iodine, that is the simple iodine solution, this iodine solution is going to react with the crystal violet, and it is going to stain the color of the cell wall as violet. So those cell walls we have which has thick layer of peptidoglycan, they are going to be retain this violet color, while those cell walls which do not have thick peptidoglycan layer, they are going to lose it. And how they are going to lose it? They are going to lose it to decolorizer. So once you stain a particular cell with the iodine solution in crystal violet, then you are going to use decolorizer. So thinner peptidoglycan wall is going to lose the crystal violet color, while the thick layer will retain the color of the crystal violet. Once you use this decolorizer, you move to the next step, and next step is the use of safranin stain. This is a secondary stain. This safranin is red in color. So now the cells which have lost their crystal violet color, they are going to take up the color of the safranin, that is, they are going to be red in color. And then you wash these slides and you observe those slides under the microscope. So this is what the result looks like. These are the cells which are going to retain their blue color, that is violet color, and these are the cells which lost their violet color to the use of decolorizer, and they took up the color of the safranin, that is red or reddish pink. So this is how these group of organisms, so these group of bacteria are having their cell wall made up of very thick peptidoglycan, which is why they are able to retain their first color, that is the violet color, whereas this group of cells or the bacteria, they do not have a thick peptidoglycan layer. Instead they have a very thin wall. That is why they lose the color of the crystal violet or the iodine's gram, and they end up taking the secondary stain color, that is this color of safranin, which is red in color. And that is why these cells or bacteria are called gram positive bacteria, and these bacteria are called gram negative bacteria. It is very pretty simple technique which we commonly used in our labs. So I hope this much is clear with you. You know that what are the basic features of a prokaryotic cell, you know what is gram staining technique, and then once you know prokaryotic features, you need to understand how is it different from a eukaryotic cell. So this is the very basically I have tried to explain. Anyway, I am going to do the eukaryotic cell in detail in my next video. For now, you need to understand very basic differences. So what we have studied till now, that a prokaryotic cell is having a cell wall, a plasma membrane, it is having a flagella for the movement, it has 5 photosynthetic membranes if it is going to synthesize its own food, then it has freely lying ribosomes which is 70S type, then it has food reserves or other inclusion bodies which are freely available in the cytoplasm. The DNA is in the form of nucleoid, not well defined, not covered with the nuclear membrane. It has pili of fimbriae which are helpful in the process of conjugation, and the cell membrane is having the infolding called mesosome, which increases the surface area of the cell membrane and helps in the process of respiration. Whereas in case of eukaryotic cell, if it is a plant, it will have a cell wall. If it is an animal cell, it will not have any cell wall. For that, you can see the first thing you need to see is the nucleus. So eukaryotic, as I said, karyon means nucleus and U means true. So true nucleus is found in a eukaryotic cell. It means the nucleus is well defined, tightly bound because of the presence of histone proteins, and well surrounded by the nuclear membrane. So therefore this is called as true nucleus. That is why it is a eukaryotic cell. Then and all the other cell organelles are covered by a well defined membrane. It has a presence of mitochondria, Golgi apparatus, and endoplasmic reticulum. So these are the things which are found in a eukaryotic cell. We will be studying each and every cell organelle in detail from the later videos. So once you are done with the pictographic way, you come to the table, and in table you need to explain the difference between a prokaryotic cell and a eukaryotic cell. The basic difference is the cell size. Okay, first of all you need to write that pro means primitive, karyon means nucleus. So here primitive nucleus is found, whereas in case of eukaryotic cell you will see the presence of a true nucleus, that is a nucleus which is tightly bound and having a well definition and also surrounded by a well defined nuclear membrane. The size is very small in case of prokaryotic cell, while in eukaryotic cell it is much bigger. The flagella is single-stranded and only one fibril is present inside. But in case of eukaryotic cell, the fibrils are present in the flagella is in the form of 9+2 pattern. Don't worry, that we will study once we will be doing the flagellum in detail in the later videos. I will explain to you what is this N9+2 pattern. The organelles like endoplasmic reticulum, Golgi apparatus, centroid, lysosomes, these are absent in prokaryotic cell while all are present in case of eukaryotic cells. The same thing, mitochondria is present in eukaryotic cell, which does the function of respiration, that is your Krebs cycle and all. In case of prokaryotic cell, mitochondria is absent. However, the process of respiration is done by this specialized structure called as mesosome, which is nothing but the infolding of the inner cell membrane. The ribosome in case of prokaryotic is 70S type, while eukaryotic both 70S type and 80S type are found. Again, I will explain what is 70S type and 80S type when we will be doing ribosome in detail. Here chromosome is present, it is naked, it means it is not tightly bound and not surrounded by a cell nuclear membrane. In case of this, multiple chromosomes are present, they are very tightly formed, well structured because of the presence of histone proteins. Here true nucleus is present with well defined nuclear membrane. Here there is no true nucleus, it means it does not have a nuclear membrane, that is called nucleoid, in which it is the region where the genetic material of a prokaryotic cell resides. The DNA is without histone proteins, that I had already told, and DNA here is present with histone proteins, which gives its unique feature of tightly bound and well structured. Thylakoid is absent. In case of thylakoid, present in granum in the plant eukaryotic cell. Sex pili is present for the conjugation process. In case of eukaryotic, it is absent. So again, I am telling you, these are the basics of prokaryotic cells. Each and every part which is present within the eukaryotic cell, that particular cell organelle we will be doing in detail in the later videos. So if in case you're not clear with any of those points which are present in eukaryotic cells, don't be worried, we'll be doing in the later videos. With this, we come to the end of today's discussion. Please take care. All the best. Thank you.